next sessions will also be recorded. And we will be sending out the recordings as well as the slides to you all with the mailing list that we sent you this link. Please ask your questions in the chat, and whichever one of us is not presenting will answer those questions to you there. Or if you have a more like discussion question, we do have about 10 minutes at the end of this talk for those larger questions. So here's our agenda for the day. We're going to talk about why we need cellular agriculture, or why it's important to study, what it is, a brief history of where it is so far. We're going to spend a large portion of this session on how exactly does it work, what's the science, and then if we have time, we're going to talk a little bit about what the future for cellular agriculture might look like. So let's get into why we need cell ag. Some of you might be vegan or vegetarian, and you might know some about this, but we're going to talk, have a brief summary of why is animal ag pretty unsafe, unethical, and unsustainable. So first, just in terms of resource use, it's basically going to be impossible to keep up the demand for meat with current animal agriculture systems. Just in terms of land use, 50% of our habitable land is already used for agriculture, and 77% of that is just for animal agriculture. So that includes both land where we're farming those animals, as well as all of the soy and corn and other crops that we could be eating ourselves, but instead are feeding these animals. And a large part of the other habitable land that we have is already for forest and shrubs, and we're seeing a huge amount of deforestation that is being used, that is being done by meat ranchers to try to have more land for their farming. So for instance, in the Amazon, you've been seeing this deforestation that's caused by farmers who are trying to use this land for beef production. Next, in terms of pollution, food production is one of our largest producers of greenhouse gas emissions, and the majority of that comes from animal agriculture, with most of that coming from beef. And then just in terms of animal welfare, a lot of the large majority of animals that end up on your plate do not actually live on those happy bucolic farms they show on brands. Instead, they're going to be living in these huge factory farms or these industrial sheds like this, where they're very cramped conditions. They're basically swimming in their own waste. They will fight with each other, and they have to be debeaked or dehorned without anesthesia. Well, they don't have to be without anesthesia, but they are, in order to try to minimize the number of fighting, the amount of fighting. In terms of foodborne illnesses, we're seeing this right now in our current COVID pandemic, as the COVID pandemic is suspected to have originated from a wet market where live whole animals were being kept in close proximity to each other and humans, and that was believed to be how that disease moved over to humans, as well as our past pandemics of the 1918 influenza, HIV, H1N1, all originated from hunted or farmed animals. So in addition to those pandemics, we're subject to just constant E. coli, salmonella, and viruses coming from the biowaste that is produced from these animals, because we don't just get meat from these animals, they're also producing manure, blood, pus, mucus, and that can spatter on the meat, or it's also been known to spill over and contaminate waterways. And this is just a map of the events that have happened in Illinois of those spills. And then in terms of antibiotic resistance, we are using a large amount of antibiotics on animals, not just to keep them from disease in those huge crowded conditions of the industrial sheds, but also because it helps them grow faster. And this large antibiotic use is causing there to be more hardy strains of microbes. So we need to come up with new harder drugs that have harder side effects on our systems. So then I just want to talk a little bit about how a traditionally farmed sausage gets made. The cow is, the animal is born. It might die before it can reach weight, but if it does reach weight, then I had decided not to go over this part to ease your stomachs, but basically it gets slaughtered, cut up into its component parts in a huge assembly line, and then otherwise processed downstream. So basically it's a huge number of steps. You're transferring a whole animal from like a little baby to a grown adult to a sausage. But even with all of that inefficiencies, we're finding it's quite hard to give up animals. A large portion of people are not vegetarian or vegans, but even those who are, we're finding abandon their diet. 84% of them abandon their diet, often quite quickly. And the main reasons for doing this is that they're unsatisfied with food, or they're seeing health problems. It's inconvenient, and there's just a lot of social factors at play because meat and other animal products are a huge part of our cultures. So we're seeing that even though we have Burger King introducing the Impossible Whopper and all this exciting news, that meat demand is actually rising, especially in growing economies like China and the rest of Asia and Africa. So we need an alternative. What is cell ag? Cellular agriculture is the production of agriculture products from cell cultures. This is a broad definition. You might see other definitions, but I think this is the broadest definition while having some sense of definement. There are two types of products that you have. You can have cultured meat, or you can have precision fermentation or acellular products. So acellular products are, you can think of the things that vegetarians would eat if you're thinking about a traditionally farmed system.
I'll have it. I will have it.
Please mute yourself if needed. Thank you. So cell ag encompasses cultivated meat, which is made from animal cells, and these fermentation products that are grown from having compounds that are traditionally found in animals inserted into microbes, and those microbes grow it up for us. This is actually done for penicillin currently, as well as some other molecules, but cellular agriculture is increasing that product range to, for instance, whey or egg white proteins. Cell ag does not encompass plant-based proteins like the Beyond Meat burger you see here, although it does actually incorporate the Impossible products, but not because Impossible is made from animal cells, but actually that Impossible uses heme grown by fermentation to color their products a nice meaty color. So the field is really growing in these past, I would say the first few companies started out only maybe 10, 15 years ago. And in the past few years, we're starting to see more of a wave of companies working on media and scaffolding and all the ingredients that you need to make cultured meat. I also did want to give a brief shout out to our USA-based nonprofits, New Harvest and the Good Food Institute, that we're probably going to talk about often in our longer intro course, and also you'll see in our seminar series. So a little history of cell ag. How did we get here? This actually isn't a isn't a new field. This is the actual application is just coming to fruition, but we started keeping muscle cells alive outside of the body in 1912. Winston Churchill actually said in 1932, he predicted that 50 years hence we shall escape this absurdity of growing a whole chicken in order to eat the breast or wing by growing these parts separately under a suitable medium. So it took a bit longer than 50 years, but we are starting to see us overcome that inefficiency and all of that suffering. So the first patent for cultured meat was made in 1999, and NASA was sort of the first supporter of these movements and thinking about making meat for astronauts in space and space travel. New Harvest was founded in 2004. Mark Post's very expensive but very exciting hamburger was made in 2013. The price dropped from $325,000 to less than $12 in 2015. Perfect Day released the first commercially available cell-ag product, which was a ice cream made with cultured whey in 2019. And the first cultured meat product was made, as we said, just last month to be served in a restaurant in Singapore.
Emily, I want to add one more thing on the timeline.
Yeah.
There's a kind of quite a big jump between like 1932 and 1999. And between that time, there was a lot of different scientific innovations that were happening.
Absolutely.
Mostly it was for not specifically cellular agriculture, but just developments in improving media, improving how we grow cells, understanding differentiation and stem cells, et cetera. They're like super important. We kind of want to highlight more of the cultured meat aspects of it.
Yeah, thank you. All right, so let's go into the science. How does the cultured meat sausage get made? So here is a brief overview of the process that we're going to really break into parts and dive into. First, you take a biopsy from the animal. You isolate the specific cell type that you want from them. So you might have like skin cells, fat, whatever in there, but you really just want the muscle stem cells. You might preserve those stem cells, but then you're going to grow them up. They differentiate into your fantastic steak or whatever you want. You harvest and process them and you serve them as meat. So first, looking at the biopsies, there are a couple of characteristics you want to keep in mind when you're taking your biopsy. You're going to have to think about where you're taking the biopsy. So the amount and quality of muscle stem cells you get is going to change whether you're taking a biopsy from the front shoulder or the back shoulder of the cow, for instance. The age of the animal is definitely going to matter. As we've seen in all mammals, your muscle stem cell concentration will decrease as you age. The breed of the animal might matter in terms of not just the concentration of muscle stem cells themselves, but also if you have any heritage breeds or like special flavors that you would like to keep. You also want to think about the sex of the animal and what are the environmental conditions the animal is being kept in. Is it having a lot of stress? Is it getting lots of exercise? Is it having a good diet? Then you want to first fixate the animal in place, give it anesthesia, and then have the biopsy. So you can do either a needle biopsy, which is just kind of like a shot where there's minimal trauma, or you can have an incision where there's it's a larger size, so there's going to be more trauma, but you might get a larger sample. And we also want to think about animal welfare as we're making this biopsies, because this is the one part where we'll still rely on a small number of animals, and we still want to minimize the suffering of these animals. So the most stressful part of this whole process for the animal will probably actually be during fixation, when it's being put into a little carrier thing and giving it an anesthetic shot. So we want to maximize the number of biopsies that we take per session, not just have one, so that we minimize those number of sessions. We let the animal recover for at least a month, and we're going to slightly vary the location that we take the biopsy so that the animal can heal in that location. And throughout this presentation, I'm going to have a number of other sources that you can check out on your free time to learn more. So next, talking about the isolation of those muscle stem cells. Learning a little bit about muscle stem cells. They're basically the adult stem cells that your muscles use to heal. They're present in skeletal muscle tissue. So they have the ability to self-renew your muscles, but they don't have the huge capacity to proliferate into whole different things like liver or blood or bone that an embryonic stem cell or ESC might have. But it has a higher proliferation capacity, or like higher ability to grow than do mature muscle cells. So when you're isolating a muscle stem cell, you have to physically disassociate it in your biopsy, then do enzymatic dissociation, filter, and then centrifuge to remove all of the debris and isolate just all of the cell types. Then you can isolate the specific cell type of muscle stem cells by FACS, MAX, density gradient centrifugation, or pre-plating. So next, you might want to preserve those muscle stem cells, especially if you're taking multiple samples at one time, or if you would like to overcome the Hayflick limit, which is that cells, that mature cells typically only go through about 50 cell divisions before they will just stop dividing, become senescent. And this is quite important for like cancer, where most cancers have broken this Hayflick limit and become tumors. But we would like there we would like to also overcome this Hayflick limit in our own tissues. So there are a couple of ways that we have for immortalizing cell lines, where we can either isolate naturally immortalized cells. So for instance, the HeLa cells, if you've ever read The Immortal Life of Henrietta Lacks, those came from a cervical cancer and were found to just keep dividing infinitely. Or you can immortalize those cells yourself by having different methods to express telomerase, which heals the DNA damage that it causes cell senescence, or otherwise inactivate the checkpoints that your cells have that cause senescence. So there is sort of a trade-off, and different companies might use a primary cell culture that has this Hayflick limit, or a cell line that has overcome this Hayflick limit. The primary cells, of course, have a limited cell capacity. There's variability between samples because you're going to take the biopsy from a different location or different count each time. It still requires animals, of course, but it's non-cancerous at least. Versus cell lines overcome a lot of these challenges, but there's still going to be an accumulation of mutations over time, though not from sample to sample. And there is going to be oncogene expression, that although this will not likely be a problem for like consumption, because the meat will of course be cooked, and your body does a good job typically of eating meat that includes cancerous cells all the time. But desired attributes of all of these cells are you want them to have your optimal taste, texture, nutrition for the consumers to make them happy. You might have a heritage breed like the Wagyu beef. You want it to be safe for consumption, of course. You want it to grow well at scale. We're thinking of like thousands of liters in a bioreactor. So that there are a lot of manipulations to cells to make them be able to grow at such a large scale. And it would also be nice for them to be non-GMO because they're finding that to be quite important for consumers. So here I'm going to leave Adil to talk about media because he is our media expert.
Thank you for that introduction. Yeah, so I'm going to go over all the components of actually kind of what you need to sustain and grow your cells. Yeah, so go next slide. So what exactly is media? Basically, it's just a solution of nutrients that are used to grow and feed cells when they are not in the body, so an in vitro environment. And media plays a multitude of different roles depending on the type that you're using and the context. But some of the key attributes it can do is also maintain the pH of the environment, improve the osmotic pressure to make sure that the cells are growing properly. It can help with proliferation of the cells and possibly even differentiation depending on the media you're using. Yeah, and so media is really important in the context of cellular agriculture because estimates right now put it at anywhere between 55 to 95% of the marginal cost of production of cultured meat. And that is like very prohibitive, and why one of the original burgers that was made back in 2013 cost over $300,000, which isn't sustainable. And I mean, since then there's been a massive amount of work that has been focused on reducing media formulations, and at least some of the key larger companies have stated they have gotten massive reductions in costs, but it's still a really important challenge for the field. How do you develop low cost, scalable media formulation so that you can actually produce bulk amount of meat? Okay, so what exactly is in media? It is we have a question. Is the cost also because all media needs to be yeah, so the GMP compliant is a component to it, but not really the main cost, and we'll kind of go into where the cost comes from for media, but the processing methodologies isn't the biggest component. So what exactly is in culture media? It includes this is GMP is like just good manufacturing practices. It's a common method and standard for how you make different materials. So you can click the next slide. Yeah, so the first thing you need is kind of having a good carbon-based energy source. Usually that is glucose. So you need glucose for your cells to grow. Next, you need amino acids. There's plenty of amino acids that are not naturally made by cells, and they need to intake them. And so because of that, you need to make sure that your culture media has a lot of good amino acids. Next one is salts. So salts are important because they can act as cofactors. Cofactors means that they are like helping out with enzymes to do specific functions, or they are in themselves improving osmotic pressure. And there's a variety of different reasons why you might need salts. The next are vitamins, again, kind of acting as cofactors to help enzymes inside of a cell do multiple different roles, but vitamins, again, kind of play a lot of different roles, including helping the cell grow in general. And then, of course, you need water, as cells are made substantially of water. And all of that together kind of composes what is generally referred to as basal media. And so that's the base components that you need to grow a cell. And then there are usually other components that can support the cell viability and vitality throughout its growth. And mainly that is serum. So you can click the next one. Yeah. So mainly that's mostly composed of serum. And so what is serum? Basically, if you took blood and you centrifuged it, you would see kind of a separation between this like clear-ish liquid at the top, and then components that kind of are composed with blood clots. And that top component is serum. So next slide. Yeah. So serum includes all the electrolytes, antibodies, antigens, other proteins, hormones, fats, and anything else that is kind of in your blood that is not red blood cells, white blood cells, platelets, or any clotting factors. It's similar to plasma, but slightly different. If you took a lot of like the clotting factors out of plasma, you would basically have serum. So yeah, and serum is at least in common methodologies for growing cells. Serum will compose anywhere between 5 to 20% of the final media that you're using. So a lot of it is going to be basal media, and then the extra bit is going to be serum. And the most commonly used serum is fetal bovine serum. So that's referred to as FBS. And FBS is a byproduct of the meat production industry. And basically, the way that FBS is acquired is that when a pregnant cow is found during the process of slaughtering, the blood is extracted from any kind of mature fetus, and that blood is then used to make FBS. So if you go to the next one. And that blood is what you do is you take that blood, you'll coagulate it usually at a low temperature, centrifuge it, similar to the slide I showed before, to get the serum out, and then that serum is analyzed for any viruses or microbes to make sure that it can be used to grow cells. And anywhere about 8% of cows during the process of slaughtering and meat processing are going to be pregnant. So you generally have a fixed supply of FBS that is made and used. Okay, and so we have some questions. So let me look at that really quickly.
I think one of them is just like, can we not use FBS, please?
Yes, that's a great question, and I'll get into that, alternatives to FBS. So that I'll definitely talk about that. So I just wanted to get into kind of the main media formulations are used and how people kind of go about doing that. And yes, FBS it's not animal free and it has a lot of ethical problems with it. Even the process for getting FBS is definitely really ethically problematic. And not just that, it's also really expensive. You have a fixed supply of FBS, and it can cost right around $1,000 per liter. And that supply that's currently out there will not be nearly enough to meet the demand if we're going to start growing cells in bulk for human consumption. And on top of that, there's just high degree of volatility in the FBS market for you to be able to acquire it, depending on the different sources that you're getting at. But and there's plenty of reasons. I'm not going to get into that, but yeah. So next is that because you are kind of harvesting FBS and it's just an organic material that's getting extracted, there's going to be batch to batch variability. So from when you harvest from one group versus when you harvest again later, there's going to be differences that make it really not really a reliable method. And then next one is FBS. So you go back quickly. Yeah, FBS is not chemically defined. So all the different components that are in FBS are not 100% clear. And because there's variability as well, we have to reanalyze it every single time. And still there's some question marks about what's there, which creates problems with reproducibility. Okay, so as a lot of people mentioned, FBS is not really the way you want to make things going forward. I kind of bring it up because it is one of the main methods that is still used and oftentimes a starting point for how for a media formulation that you'll use to grow cells. And the question is like serum necessary? It's actually quite important if you want to grow cells for a long period of time, but there are alternatives. So click the next slide. It's a lot of scientists, specifically working in the 70s, were really interested in, okay, what are actually the chemical components that are essential for you to grow a cell and for a long period of time and sustained, and they can figure out which are the components that can replace serum. And this is an example of a commonly used serum-free formulation called Essential 8, which kind of has like these 8 main components. Basal media, which we talked about before. So basal media is not actually one component. It's made up of like 50 or so components that we kind of talked about earlier, like the carbon sources, amino acids, etc. And then a couple of different salts, and then growth factors that are used. Yeah, and if you kind of do a deep dive into serum-free media formulation, there's actually just 2 main growth factors that will be contributing like 96% of the cost. So I also want to note that if you look at it already, so this serum-free medium formulation, this is an example, a great paper that does a deep dive into different components of Essential 8. So again, Essential 8 isn't the only serum-free medium formulation, but it's a good example. And at scale, currently, if you tried to make it, it would cost like $376, which is already substantially reduced from FBS, but FBS is usually just a percentage of media formulations in general. But it is quite cheap in general, but you would need it to be much, much cheaper for it to be produced. And so kind of when you look at the deep dive, there's these 2 growth factors, FGF2 and TGF beta, that contribute most of the costs. And the reason why they are so expensive is because the main method for how they're produced is by using mammalian cells to grow and make these different proteins. So that's a really expensive process. And even you can look at this. This is at scale. So when you talk about at scale, it's usually like 20,000 liters that are going to be produced at a time. And even then, you only need 0.04 grams of like TGF beta, and that is already netting you like $80 million. So these are very expensive components that are needed at really small quantities and contribute a lot to media costs. So you go next slide. So now I'm kind of listing off a bunch of different serum-free media formulations, M-Tesser, Essential 8, different versions of them, and they all kind of hover around between $400 and $600. And for the past like 20 years, people have been working on using and improving these different media formulations. And if you went out and tried to use serum-free media formulations, going to like Thermo Fisher or Gibbon, you would get one of these. So next slide. But there's a lot of research that's been done to actually reduce the cost substantially of serum-free media formulation. This paper is from Paul Burridge from Northwestern, who is working on scaled production of stem cells, specifically works in therapeutics. But the way that the first kind of cost reduction is done here is by actually just producing those 2 growth factors that I mentioned that were commonly made in mammalian cells by using E. coli. And you can figure out how to mutate them specifically to be able to produce produce them at scale. And one of the main reasons why people didn't do that before was just that these growth factors don't work super well, or they have specific like post-translational modifications that make them difficult to make. And if you go to the next slide, so it kind of shows that again, there was another cost reduction that comes from reducing the amount of growth factors that are needed and producing it at scale. So it kind of gets you at $11 per liter, which is the one of the lowest that you'll see in academic papers that show sustained growth of stem cells. Yeah, so. But again, this is not the only place you can do cost reductions, and this is again from a report on a report on the serum-free medium formulations that was done that kind of suggested a bunch of interesting strategies of reducing the costs to not just get to $11 per liter. So even at $11 per liter, it is cost prohibitive for you to be making meat. You need to get down to like $1 or less, right? Actually, really, you want to get much lower than $1 for you to be producing cultured meat at scale to be cost competitive with current meat that's produced. And a lot of the ways that that's done is really focusing on those main components that are contributing to costs. And the first one is kind of how do you adjust your cell lines to not need as much media, and then how do you reduce the growth factors by making it through E. coli and producing it at scale, and then kind of diving into the basal media as well now, and seeing what are the components that are contributing a lot to the costs. And a lot of that is buffers and different salts, and finding kind of food grade alternatives instead of medical grade, which is what's commonly used. Gets you down to right about where you would need to be in this theoretical analysis. But this is not the only methodology, and definitely not the only one that's being explored. Another main way that people are looking into how you make serum-free medium formulations, and specifically the main cost-driving growth factors, is looking at yeast extracts, algal extracts, fungal components, figuring out how to make growth factors using plant-based materials, et cetera, that can be made at scale, really, and have a lower price point. And engineering cell lines to reduce costs, trying to figure out how you can replace some of the growth factors by using what's called feeder cells. So a cell that is used to produce the growth factors for another cell, where the original cell doesn't need the growth factors to be able to grow. And then media recycling, which is basically a process of reusing media that you've already used to grow cells to kind of get the most out of it. Yeah, so that was definitely kind of a dive into that. And let me look at there's some questions.
We can answer the questions at the end, but I think
Okay.
we answered all of them. Cool. Thank you.
Yeah.
Yeah, so it's going to be back to me to talk about scaffolds, which are important for like steps 4 through 7 of this diagram, depending on whether or not the scaffolds are part of your final product. So scaffolds provide structure and support for the cells. So they're going to help mimic the extracellular matrix for the cells, and give geometric cues for correct differentiation. It can actually be used to also help offset that cost of all of those different factors that Adil talked about, where instead of introducing these expensive chemical cues, you can introduce geometric cues to some extent to reduce that media cost. It's also important for consumers because actually that extracellular matrix is a large part of the structure in your structured meat, like a steak or a pork chop, although not so important for your more processed meat like a sausage or a hot dog. And when you're talking about scaffolds, you can either have edible scaffolds, you can have scaffolds that degrade before they are before the meat is made into its consumer product, or you can have scaffolds that detach from the cells during the processing part of during the downstream processing of cultured meat, and can be reused in later batches. So when you're talking about scaffolds, you can either have scaffolds made from animal proteins that are produced by fermentation, which we're going to talk about later. So these are what is actually used in your extracellular matrix inside animals, but it can be produced through fermentation. So collagen, fibronectin, laminin. You can also use silk fibrilin, elastin, to give that support. You can also use plant or fungi proteins like chitin or cellulose or alginate, even TVP, if you've heard of that meat alternative. It can also be used to grow meat. Or you can talk about synthetic scaffolds like PLGA or PCL that would be more reusable, not edible. So I am skipping over a lot of science because we're going to be talking about this topic in our later intro sessions. But I will say that you're going to need different scaffolds for muscle cells than for fat cells, which are both going to be important cell types for your final meat product. Actually, the first 2013 hamburger lacked fat cells, and so the taste testers noted over and over again that it lacked the juiciness of a hamburger because it didn't have those fat cells. So to make those fat cells, you're going to actually need a different scaffold, although you're going to use a similar isolation process that we saw in steps 2 that I talked about earlier. So basically, like the adipocytes or your fat cells are going to like microbead scaffolds better than the aligned scaffolds that your muscle cells like. And then you have different ways of combining those two scaffolds together that the Roat lab in the UC system is working on right now. So then I want to talk about a couple of ways that you want to optimize your scaffolds. You want to optimize the rate at which your cells are attaching to your scaffold, so you're not getting this inefficiency that you have a bunch of cells, but they're not even growing into a larger tissue structure because they're not attaching to the scaffold. You also want to make sure your scaffold maximizes your cell growth rate, and it has this optimal degradation rate if it does need to be degraded, where it won't degrade during the time where it's being used to culture your cells, but it will degrade either before processing, or it will degrade in your body if it's edible. So I'll go over 2 ways that you can make these scaffolds. The first one is 3D printing, which is being used with a project funded by New Harvest, as well as by 2 of the companies in the space, Novameat and Blue Nalu, where you're basically just 3D printing a structure that has your required geometry, and then cross-linking that to make a biolink. Or you can actually use something in your own backyard. So researchers in the Gaudet lab at WPI have actually shown that you can decellularize or like take the cells out of leaves or apples or any other plant material, and then inject animal cells into them. And this method is quite useful because leaves have natural vascularization that allow for the diffusion of oxygen and other nutrients into those cells that are important. Here's more resources if you want to learn more about scaffolds. There's a lot of papers on this topic. Next, I want to go into bioreactors. So bioreactors are the chambers that you use to control cell growth. When we're talking about small scale, for research purposes, you're probably just going to be using just small lab flasks to grow your cells. But once you're producing meat at scale, you really want to reduce the manual labor costs. So you're going to be using bioreactors, which are automated and controlled to keep those cells at their ideal nutrient levels, temperature, etc., and at a ideal schedule without huge manual labor costs. So there are a few different types of ways to categorize bioreactors. One of them is whether you're going to have a single-use bioreactor or it's going to be stainless steel. So single use is quite popular right now. These are bioreactors made out of plastic. They're popular because they don't require heated sterilization, so they have a faster turnaround time, and they don't have that water energy cost that is required when you need to sterilize between batches. But it is more expensive long-term because you have to throw the bioreactor away after its use, so there's a huge amount of waste as well. Versus stainless steel bioreactors like you saw over there, or like you see in your craft brewery, if you've ever been to one. These require heated sterilization, so that takes time to clean up, but it's less expensive long-term and it doesn't have that plastic waste. So then you also have a category of how is media and nutrients flowing through your bioreactor. So you can have an agitated bioreactor or a perfusion bioreactor. So agitated is that you're mixing the media in some way. The most common, easy one to picture is through stirring, where you just have that paddle, as you see here, that stirs the media around. And this is mostly for a batch bioreactor where like you just have a set amount of media and then you finish, you take the cells out. So in contrast to agitated media, you can have a perfusion bioreactor, which is when you have a steady flow of media over a cell population. So this can be a more continuous process where you're having a continuous flow of media. So one example of a bioreactor that is being worked on by Scott Allen at the University of Bath is this hollow fiber membrane reactor, where your scaffold is actually this porous straw that you see here. And waste and medium is circulating through these straws and perfusing out of the pores in the straws in the hollow fiber out to the cells. And this bioreactor is especially impressive because you can have a quite high cell density while still allowing your cells to get access to the media that they need. So when you talk about media, about your bioreactor, you have to think about this cell density. So like how much space do you need to grow a given amount of meat? You really want to maximize the time that you're in exponential cell growth, which is like the fastest cell growth that is possible for the cells. You want to minimize that space requirement, minimize your energy requirements, and have less consumables. And of course, you also want to reduce your labor costs, so you want it to be automated. In addition to reducing labor costs, that will also, of course, reduce the possibility for contamination, because contamination will most likely come from when you have to like manually touch the cells or have them exposed to air. So we actually have 3 companies already that have started their pilot plans for culture meat. Supermeat, Eat Just, Biotech Foods have all already have their pilot plans. There are about a handful of other companies that are building their pilot plants now after completing their fundraising needed for it. And there's also a whole bunch of other papers that you can dive into this topic as well. I'll be talking a lot more about the different types of bioreactors in our later presentations.
I also wanted to add one more thing to the bioreactors component. I think it's really important to also realize, like, if you're talking about like substantially replacing a good percentage of the way we eat meats or really other types of foods, including other fermented products that we'll talk to you about later, you need very large facilities to accomplish this. And like currently the facilities that are out there that kind of have these types of bioreactor designs are majority focused on pharmaceuticals. And even then they're quite small because the volumes that you need are really small. So a lot of work needs to kind of be put into innovating in how you make these facilities at scale, at low cost, and understanding kind of what are the cost limitations and infrastructure necessary to be able to do this well and at scale, especially when you talk about making it in different parts of the world.
All right, so then I'm going to have a brief look into downstream processing. Unfortunately, there aren't a lot of papers available on this topic that I was able to find. But here is a diagram of basically how downstream processing would work, depending on whether you have an edible scaffold that's going to be part of your final product, or you're going to have an inedible scaffold. So if you had an inedible scaffold, you're going to have to detach your cells from that scaffold once they are done using it. And you can detach it with, for instance, TRPLE, which is an enzyme mixture, and then centrifuge it, the cells out, filter the cells, then press the cells together into a tissue and harvest it. The process is a bit simpler if you have an edible scaffold. You basically just also drain the media out of the bioreactor, press the tissues together and harvest. So now we've gone an overview of how cultured meat is made. I want to talk about safety. So at least in the US, safety is going to be regulated jointly by the FDA, or the Food Drug Administration, and the USDA, the Department of Agriculture. So the FDA is going to be regulating the parts of cultured meat from the biopsy to when we harvest those cells from the bioreactor. As these processes are basically similar to processes that the FDA already regulates, for instance, with drugs or pharmaceuticals that we've seen. But the USDA is more interested in regulating food processing. How will this be labeled as steak or hamburgers, for instance? So that's going to be the regulations framework for meat, catfish, eggs. And the FDA is going to be in control of the entire process for seafood, as FDA is in control of also like farmed seafood currently. So then another big question is, how will cultured meat taste, and will it taste as it should? When we think about how meat tastes, that flavor is going to come from a unique mixture of the fat cells, the fatty acids, in both cells and outside of them, the muscle cells or the fibroblasts, as well as collagen and different flavor compounds. So we really need to make sure that we're going to mimic that whole array of compounds and cells in order to get the true meaty taste. But so far, since that 2013 not so juicy hamburger, now that we've added fat cells in, it turns out that the flavor tests have come back pretty positive. So taste testers have said it tastes like chicken. It breaks, it flakes, it's extremely tender. It has a pleasant meaty flavor and is achieving the typical meat bite or texture, if you want to talk more professionally, which is very exciting. And then we can not just expand to how meat typically tastes or the typical nutrition profile of meat. One of the exciting things about cultured meat is that you're not prohibited to what is possible in just thinking about animals. So you can think about enhanced meat, where, for instance, in Andrew Stout's research at the Kaplan Lab, he is enhancing the nutritional profile of his meat so that there's a lot of vitamin A, which is they're targeting for a elderly audience where they don't have to remember to take a pill. They can just eat their normal steak for dinner and they have their vitamins. Or you can also culture the meat of animals that you would not typically be able to farm. So for instance, here is a dumpling made from kangaroo meat. All right, so I have talked a lot. We're going to have a 3-minute break where people can run to the restroom, take a drink of water, and then we're going to come back to talk about fermentation products or acellular products with Sangeeta. Thank you. Or you're inserting a new gene in. So that might be a GMO product. Ways to get around that are to use naturally immortalized cells or to actually have footprint-free methods of genetic modification where those cells are initially genetically modified, like you've knocked out a gene, but there's actually methods where you can add another chemical or biological cue, and those genetic modifications are removed when you before they are turned into the final product that a consumer would see or consume.
Yeah, I think like just the most simplest kind of version of that is really if you try to make pretty much any modification to your cell, it can result in potentially being labeled as GMO. So, like the strategy that probably is is very unlikely for it to necessarily be labeled as GMO is if you take a biopsy, so you have a good primary cell, and you use it up to the point that it can you let the cell grow and split as much as it possibly can, hitting its Hayflick limit, and just use that to grow.
Yeah,
the
primary cultures.
Yeah, at any time if you try to like immortalize your cell or convert it into like an IPS potentially, or try to go more exotic and add things like trying to add like beta carotene or there's a lot of different strategies people are looking at to kind of improve the nutritional profile of meat. This is especially true if you look at Memphis Meats, which is one of the leading culture meat companies. All of those things, I think you'll basically get labeled as GMO. Kind of have my own thoughts about whether or not GMOs are bad, but at least in terms of consumer acceptance, it's it's important to note these things.
Yeah. So, and then the other question is, what is a common example of a consumable? So a common example is the single-use bioreactor themselves would be a huge consumable, as well as the media that Adil talked about, where there is a lot of technology going into how can we recycle that media and minimize the waste of it. How can we get every last bit of that quite expensive substance? Yeah. So I want to make sure we have time for all of Sangeeta's presentation, but we will have more time for questions at the end of this talk. So please leave your questions for the end, or we'll answer them in the chat. Thank you.
All right, can everyone see my screen with the wonderful fermentation products on it? All right, cool. So to kind of continue this conversation about GMOs and genetically modifying ingredients, let's switch gears from cultured meat and talk about the other category of cell ag, which is fermentation. And I guess one of the biggest things I want to highlight right off the bat is that fermentation is very diverse in what it can create. So in cultured meat, you're really thinking about, hey, we have these meat products and we want to recreate them. Fermentation, we can do that, and we can make a whole variety of other products as well. And just on the screen, not all of these things we would consider fermentation in the biological sense, but in the alt proteins or cell ag space, we're actually going to talk about many different types of these things. So fermentation, just as a biological definition, is the process an organism can do to create energy and grow in the absence of oxygen. But in our alternative protein space, we're going to be talking about 3 types of fermentation. So the very first one is just probably one you're very familiar with, the thing you see going on in cheese and kimchi. We're essentially just taking a microorganism, we're putting in our food, and it starts to grow within the food and transform the food properties. Another fermentation class that's pretty important in alt proteins is called biomass fermentation. And what's actually going here on this picture, this is actually not a steak. This is a mushroom fillet that was grown by a company that is trying to create meat alternatives out of mushroom mass, and their name is Meaty Foods. So we call this mycoprotein. It's actually just mushroom, so it's been flavored and processed, and it tastes and feels like meat in your mouth, but it's not actually meat. So that's not what we consider cell ag. But really, when we're talking cell ag, we're talking about this category called precision fermentation. And I put a photo here of a whey protein because precision fermentation, what we're making are these exact proteins or the exact compounds that we want to have from animals, but we're just doing it with microbes instead. And I think John earlier was asking about growth factors, if there was a way we could create these in the lab. And Adil talked about how sometimes it's a little bit harder to create these out of microbes because these growth factors like FGF2 are pretty complex, but this is a great place to start if we want to think about the concept. So how do you actually make something with precision fermentation? Somebody had brought up the name Perfect Day in the chat a little bit earlier. And what they do is they create animal-free milk. So right here we have our typical traditional process for creating milk. We have a cow, we make the milk, and in that milk is the milk proteins that we like. With precision fermentation, all we're doing is we're taking this yeast, which has been genetically engineered to be able to create this whey and the casein proteins that we really know and like in our milk. We're letting those yeasts grow and do their yeastly functions, but they also create whey. And that whey is exactly the same as what comes out of the cow. We're just doing it without having to milk a cow. Now you're going to learn how to do it too. So first we start off with yeast cells. These can also be bacteria, but you're going to have to do a few modifications, and between microorganisms can change a little bit. Most commonly and very often used is yeast for this product. And inside of the yeast cells is DNA. So for those who are not maybe coming from a science background, DNA is the code of life, and it goes through a process that can eventually become something called mRNA. And mRNA becomes something called a protein. And that protein could be whey, it could be a lot of things that we really know and love. But going back to the DNA itself, one of the most important parts that we see right here is called the coding sequence. And whatever we put in that coding sequence, we can create. So here we know something really interesting. We know how a cow can make its whey proteins. We know exactly how it works. But even more interesting, we know how we can make yeast make the exact same protein that a cow makes. And so if I was just to substitute that coding sequence for a whey protein and make a few more changes to make this go a little bit more smoothly in yeast, and voila, I have yeast that can make a whey protein. And you can make a lot of different types of proteins in this way. And this is pretty much the core of precision fermentation. So I think we referenced earlier as well, the heme protein from Impossible Foods, the hemoglobin. And originally what Impossible was doing to try to get this kind of bloody color, bloody flavor, the iron sort of tasting aroma in their Impossible burger, which is kind of shown here. What they were doing is they actually found that soy contains hemoglobin and they were kind of extracting it from the soy itself. But they found that to be kind of unsustainable when they had to actually dig up these soy plants and create soil erosion and carbon emissions. So what we do instead, they thought, is let's extract the DNA from the soy and we'll put it in yeast and grow it up. And that is exactly what you're eating if you ever have tried an Impossible burger. So that is thanks to precision fermentation. So coming back to thinking about this in a little bit of deeper scale, where are some parts within the whole precision fermentation process that we could possibly tap into more of its potential, innovate in the future? So this is kind of a chart that was published by GFI, the Good Food Institute, in their fermentation report. And we're going to kind of go through it step by step and see some cool features and improvements we could make.
Sangeeta, I just want to quickly add on what you're saying, because I think one good example that we often talk about as well is insulin. And so that process of like basically taking that DNA sequence and then putting it in, like Sangeeta was showing with whey, is how you really make insulin. You take the human insulin sequence, you put into a microbe with the same sequence, a little bit of modifications, and then produce it at bulk. So it's the same idea. This technique is quite old one. I think pioneered
Definitely.
in the 70s, I think. So it's yeah, just wanted to mention that.
Yeah, absolutely. Adil is completely right in saying that this is not a new technique at all. And in that sense, one of the kind of goals we hope you guys are able to take away from this presentation and this part of the presentation is what GMOs are and how they came to be. And hopefully you can help mitigate some confusion or fear that might come about it from other people. So along with insulin is actually one of the older proteins that was created through fermentation is known as chymosin, which is actually the thing that helps curdle your cheese in milk. So we know how to make a protein, be it whey, heme, chymosin, insulin even. What if we wanted to make a chemical? A chemical has no amino acids or protein components inside of it. So this is vitamin B12, which it's found in red meat a lot, and sometimes non-meat eaters take supplements for it. One way you could make vitamin B12, you could have 91 postdoctoral researchers from both Harvard and ETH over the span of about 12 years and 19 different countries work on trying to create this intensely complex chemical reaction to create it. Or we could find out that yeast actually makes this in a relatively simple process called biosynthesis using enzymes. And so instead of using this whole chemical process, we could have our yeast just create these enzymes and those proteins can make the B12 for us, saving a lot of chemicals, a lot of time. The other sort of interesting thing with choosing a target, and the third sort of interesting possibility about precision fermentation is we can essentially create anything we want. And I kind of hinted at this before, but a company called Deltor, which Emily actually interned at previously, they've been able to looking at collagen of different species, even those that we may never have had contact with. So they've been able to recreate collagen from the mastodon, as well as jellyfish, and place them into a gummy bear form, just to show people hey, this is something we can do. This is something we can achieve with this kind of technology. So taken together, in the very first step of precision fermentation, which we're really just looking at what kind of target we want and what we want to come out of this product, we'll have to think about the host. The host is a very important consideration. We want a host that can be very generationally stable, which means that over time, we're going to get consistent results in what they can produce and how they produce it. So E. coli and yeasts are two hosts we talked about and are very commonly used, but there are many other strains that are coming about and other tools in biology that are pretty recent, like high throughputs, so basically running thousands and thousands of tests and seeing which one works the best, as well as advances in synthetic biology can help us kind of isolate which hosts might be the most suitable for a particular purpose. Our next consideration, our sort of map down here, is the media. What exactly are we trying to feed these cultures to live? And so unlike cultured meat, we're not going to need a super stringent, these microbes don't have very stringent needs of serum. They're in fact very resilient and very adaptable. So for example, E. coli right here is typically heterotrophic, so it cannot make its own food. But using a technique that is kind of known as directed evolution, we can slowly coerce the E. coli to sort of take on CO2 as its primary source of energy and start to be able to become autotrophic, capable of making its own food. So the kinds of like metabolic flexibility that we're able to put into these organisms could be a really valuable thing in that we could start to see new types of feedstocks or new types of things we can feed our materials in order to create the things we actually want. And there's actually a company called Air Protein, very new technology that they're looking at into called hydrogenotrophs. So these are microbes that actually metabolize hydrogen gas as their primary source of energy. And what they're doing is they take these hydrogenotrophs, give them the genetic information to be able to create soy protein, and that's their final product. So very, very novel things that could come about the space and definitely lots of room for innovation. Okay, and the next kind of consideration we want to talk about is the bioreactor and how we could actually scale up production. So again, unlike animal cells that are growing in cultured meat, microbes don't necessarily have to have something to tether onto, any sort of scaffold for it to grow. So we can actually just put them in a huge vat of media that has sufficient circulation, and they can do very well there. They can float freely and still thrive. So this kind of bioreactor is typically known as submerged fermentation. The process is submerged fermentation. And you see this a lot actually where people are creating ethanol. A company called White Dog Labs actually just very recently bought an ethanol production facility, and they're starting to transform it into an alternative protein generation plant. And so those kind of technologies are very interchangeable. You can retrofit one to become more of the other. And in that, we're making more sustainable plays into how we can actually see this play out as a feasible reality. And I think in scale up, there's kind of a unique part of fermentation that is not talked about as much. And that's something that is called the carcass balancing problem, according to GFI. And so one way to think about this is the slaughter of a single cow can produce around 28 T-bone steaks. And so if you want to order the 29th T-bone steak, you'll have to drive the slaughter of an entire additional cow. In fermentation or in cell-based meat, we don't have to do that. We can actually say, hey, if 90% of our customers really like the more tender cuts of meat or the more tender products, we can produce 90% of our products to be exactly that, the exact ratio we need. And in that way, we minimize a lot of the waste and a lot of the resources that are going towards producing food. There's not really as much carcass to balance. So that's just kind of one of the benefits of this kind of technology in terms of both the volume that we're producing and how much of it we can produce at one time. Okay, and the last sort of step on this framework is looking at how we can actually take whatever we've grown in our microbes and turn that into our final food. And the cool thing is that a lot of microbes we find in our foods already, so it doesn't necessarily have to be something we're worried of having in our food. If we have yeast produce our whey, we can break up the yeast cells at the end of the process and not have to worry about too much about certain fragments of the yeast being in the final food. And it also depends a lot on who's our next customer. If it's a B2C company, we don't maybe have to worry so much about the post-processing steps, whereas a B2B company, which is just selling to another business, they might be doing another sort of processing step, which we don't need to worry about too much in our end. But if we're selling directly to customers, we might want to have it in its final form by incorporating other structuring, other flavorings, and other things like the Impossible Burger. And two kinds of considerations that can maybe even optimize this part of the process even further is if we can get the concentration or the inclusion level of our fermented product to be really low in our final product, that'll obviously mean we can produce more of this final product. So just for context, I think the Impossible Burger is about 2% heme by weight. And if we can get that even lower and get the same effect, that'd be a great thing. And along with that is purification. So if it's a very intense purification process, we might not be able to produce as much as we could if it was a less intensive one. So after all of the fermentation is said and done, how do we actually legally add them into our foods? So cultured meat is a very, very novel product and requires a bit of a different framework, but Perfect Day and Impossible, which are two precision fermentation ingredients, they've been added to their products, have been approved and are currently in markets. So how do they do this a little bit quicker? There's a certain classification known as GRAS, or generally recognized as safe. And the major difference between what cultured meat will have to go through versus what these precision fermentation products have gone through is that GRAS is not directly certified by the FDA. So they actually can go out and self-affirm, which means that they can meet a third party. They'll run all the tests and all of the investigations, make sure there's sufficient data to actually warrant this being safe to put in people's foods. Because, the way that we're eating in Perfect Day milk or the heme that we're getting from Impossible, it's very similar and the same to the products that we conventionally eat, when we drink milk versus when we eat meat. It's just the process that we use to get to these final products in our fermentation products is a little bit different. And that's really what needs the most look over. And so, with that being done and that kind of affirmation being done, it can be sold in market, and the FDA can degrass, of course, but they don't need to necessarily be there for all steps of the process. And so, I think when it comes to the consumer's end, we're still trying to navigate the best way that we can tell people about our products without freaking them out immediately. But that's kind of something we hope to do with this class. And I think bringing Cell Ag into more of the public eye in general, we'll hope to do is be realistic and rational about what's in our products, but make it also not something people should fear at the very beginning. And so, for example, the first kind of banner here, Microflora, is the way that Perfect Day is advertising the way they create their animal-free whey. Quorn's Mycoprotein is the way that they choose to market their protein grown from a fungus. And both of these things, they take a lot of thought and a lot of just insight from different groups of people to kind of make this dream a reality and make it something that people are willing to learn about. And so there are many, many spaces for different minds to come in and make lab-grown meat, milk, and eggs not as scary as they seem maybe at first. So with that, the revolution is here. Fermentation is a very, very, very high potential technology that has been used to make a lot of things, including animal-free milk, collagen, pet food by a brand called Wild Earth. Clara Foods has made egg whites. And Atlas is another fungi company that has made some really cool plant-based bacon or fungi-based bacon. And here are some very good resources on fermentation, the stellar of which is probably GFI's State of the Industry report on fermentation. So I really suggest you check that one out. All right.
It's back to me. It looks like we do have time to talk a little bit about what the future of cell ag might look like. So we talked a bit about how it's the generally assumed knowledge that cell ag will need to grow up to very large scales in order to increase, in order to really get that price down and have the maximum efficiency. So this is what we think about as a centralized system where you have just a few like very large factories or production units where the majority of your meat is produced. However, there are a couple of alternative scenarios that could also happen with cell ag. For instance, you could have a distributed network where you have a number of smaller factories that have less of that scale, but there has been a small number of cell ag companies that are investing in this. So there's a company called Future Meat Tech, which is patenting a small scale perfusion bioreactor, which is the type of bioreactor we talked about earlier, that can recycle media so that, and their system requires cheaper media and looks to maximize the cell concentrations compared to other types of systems. So they're hoping to decrease the need for scale and have it be a more democratic distribution of cell ag production. So then you could have distributed where a certain company owns the IP, or you could have local craft where that IP is owned by whoever is in whoever is owning a particular factory or brewery. So think about your craft beers versus Budweiser. And this picture of local craft comes from a book called the In Vitro Meat Cookbook, and it's supposed to be showing a carnery, or imagine your craft meat, your craft meadery of the future compared to your craft brewery. Or what would be really exciting is if we had a DIY system where you could actually produce your own meat at home. And this is much more futuristic. There is a group working on this sort of technology right now called Trojan Meats, which actually has these sort of recipes that people are using to figure out how they can grow their own cells using media ingredients and bioreactors that they can take from parts that they can buy online or just in your average supermarket. That's really exciting. But first we'll probably see centralization. And that centralization might be in the hands of meat processors. Like Tyson, we've seen a number of cell ag companies that are founded by current meat producers like Tyson or Smithfield in order to just like add on to their production. They see the value that cultured meat can give. Next. We can also think that the future of cell ag might be in the hands of farmers, where we actually give the farmers the bioreactors that they need to supplement their production. And this also has a lot of precedence as well, that farmers have been pretty quick to adopt new technologies, such as solar panels or even tractors have just been around for a few decades. So they might soon have their own bioreactors. Next. Or it might be in the hands of the community. Like we might have our own biomaker spaces where you could produce your meat for yourself in a common community environment where there are trained professionals that monitor you. There are a lot of different possibilities. So next. It could be in the hands of the family, like that DIY system that I showed earlier. These scenarios come from focus groups organized by Cor Vanderweel at Wageningen University. And one scenario that she came up with these focus groups of consumers was that the idea of having a pig in your backyard, that was sort of like the family pet, and you just like got cells from them once in a while. Honestly was much, yeah, cell-based meat. You got cell-based meat from them once in a while, was honestly much more, consumers could wrap their minds around that as being more natural and a more harmonious way to produce meat than both the current system of industrial animal agriculture or in the hands of meat processors at a huge scale. So that is, here are some resources for the more social science side of cell ag, or how it might look like in the future. I think that we're ready for questions and discussion. So yeah, this is a question that is quite important right now. What do you think cell ag products should be called? That is honest, attractive. I mean, we say cultured meat, and that's sort of the term that we go for. But precision fermentation is quite a new term for me. I'm not sure I like it. What do you think? Yeah, cell-based meat. Yeah, but I mean, cell-based meat is.
I think
I would like to hear you guys talk about it more, because yeah, my problem with cell-based meat is that I want it to be a specific, a term that is able to be specific as well as honest and attractive. So cell-based meat is like, well, our plant, is your pork chop right now not made of cells?
I think Ryan's comment was from a previous question, maybe.
Okay.
Yeah, sorry, I can clarify. I was just curious. GRAS seemed to cut out some regulatory hurdles for what's the right term? Precision fermentation, right? For impossible. But I'm curious if that could still apply for cell-based, or because it seems like farmers might not be really excited about cell-based meat entering the scene. So there could be, at least in the U.S., some like regulatory challenges or maybe some lobbying.
Yeah. So cell-based meat does not, or cultured meat cannot fall under the same GRAS framework, or it is not currently fallen under the same GRAS framework, and the FDA and USDA are working together to figure out a new regulatory framework for cultured meat. There are definitely a lot of precedents from current industries like pharma or fermentation that have existed for how that regulatory network might be in place, but there are new challenges to cultured meat that you have to think about. So you have to have, for instance, like allergen testing, or making sure that all of the scaffolding and growth factor and metabolites from the process are taken out after processing, but before it can be, before it is consumed. You want to make sure that you validate the cell identity, and you want to make sure that there's not microbial contamination. So
Yeah.
it is going to be a sort of new process that is a sort of a hybrid of biopharma and conventional meat regulation.
Yeah, to add on to that, I think the three types of regulation I sort of talked on were specifically for food additives. So anything you're adding in very small quantity to an existing food. Yeah, I believe that in Singapore, they actually had to come up with new framework for novel food products in order to see the just chicken to become regulated? So,
Yeah.
a great question.
And I also want to just mention, because I think we didn't go into as much detail about it, but depending on your definition, cellular agriculture can include kind of anything that's made, any products that are made that are not using the animals themselves. So that includes kind of leathers or wool, a varieties of other materials that actually have quite a similar process for production. And that's still like also it's also a really interesting space that's having a lot of momentum and work that's being done that we didn't really touch on. So if you guys are interested in that, I think there's we could talk about that as well. But
Yeah, even that. So we talked about Geltor. Geltor right now is actually not selling any products for food. They are selling products for the beauty industry. So they're selling a collagen, and people do eat collagen in their day-to-day life. We like consume it for medicine, or we have it in our meat. But they've decided for right now that they're first going to come to market in the beauty industry, because that has less stringent regulation requirements. So each time that they go through this process of regulation, there's a lot of money in order to prove that their product is safe, and the testing is less expensive for beauty. So they're going to they've come to market for beauty first, and I think have had quite a lot of success in Korea, which is sort of like a big boom, having a big boom in skincare right now. And they're moving over to now going into like nutraceuticals, like collagen pills, or looking at being incorporated into scaffolding for cultured meat and other foods.
Yeah, I was also going to ask, get a sense of how many people are interested in like entrepreneurship and working on actually like solutions that will actually reach the market versus kind of people who are more interested in just the scientific aspects and research components to it. So I don't know if there's a good way of like putting a poll, but perhaps by a show of reactions. How many people are more interested in kind of the entrepreneurship side?
I think on participants, you can do like a clap versus a like.
Oh yeah. Okay.
You can yes or no in participants too.
Oh, okay. Let's try that. Okay, yeah. So yeah, can you do a yes if you're interested in the kind of entrepreneurship, and maybe a no if you're more interested in the science? I feel like that puts a bias on it, but
I actually have a question with respect to that. Given the current state of the industry, is it would you say it's fair to say that if you're interested more in the science, you would probably end up in some big R&D group more likely than a startup, which the startups right now are more so just solely focusing on bringing things to market or sort of ideating? Or is it the case that a lot of the startups actually have some exciting science going on themselves?
Yeah, I would say actually that most of this is being done by the companies themselves. Actually, one of the things that was difficult about making this is that most of the scientific research is quite hush-hush because it's being done by companies. So most companies are doing the entire process themselves, from making the cell lines to designing the bioreactors and having that pilot plant. So I would actually recommend going to working at Memphis Meats or Masa Meat for a while if you want to get more of a sense of how that science is going. New Harvest and the Good Food Institute have done great work in funding more academic research on trying to build just like the foundation for cellular agriculture and have that foundation be open source. But yeah, if you want to really see every single part of the process, then you would probably more work for a company because academic projects, they're going to be quite specialized. So Andrew Stout is working on specifically like how can you put this vitamin A network inside a cell line, or how can you design this one specific scaffold, and hopefully it might be used for cultured meat.
Yeah, I really agree a lot with what Emily just said. I think it's fun. It's good to see like a lot of people are interested in kind of more entrepreneurship stuff, and there are definitely some component of people are just like mostly focused on the science. So I like talking about kind of where the interesting new opportunities are and where the challenges are. So I kind of wanted to mention some of those while we still have people around. So if we kind of look into the space of cellular agriculture as a whole, there's kind of the cultured meat aspects, and then there's the fermentation aspects. I'll say that fermentation is like relatively older technology that has kind of started out, as you're mentioning, like in the 70s, where people were just figuring out how to put insulin or other components into cells and then grow them up in bulk. So there's a lot of, but it kind of just stayed in that space, and not too many people actually continued on and tried to make a bunch more products during this method. But there were definitely a bunch of different people who tried different things, and there are kind of minor industries that have been developed around all of those. So there's a lot of companies and big facilities that work on food additives or enzymes that are made at industrial scales for a variety of different applications. That is kind of more of a mature market, and hasn't necessarily been applied as much for the space of food outside of some food additives or animal feeds, etc., but not like food that most people eat on a daily basis. So if you talk about interesting opportunities, I think the space of fermentation is very exciting, because right now it's kind of a land grab that's happening. So if you look at, like, for example, I think people started noticing this as like this kind of foray into just like grabbing new spaces happened somewhere in the mid 2010s. So like I think 2014, 2015 was when Perfect Day and Clara Foods, Perfect Day kind of works on milks, Clara Foods works on eggs. So where people are noticing that a lot of the foods that people eat on a daily basis that are animal based actually have a large protein component, and that if you can identify them and show that you can produce it and patent it quickly, then then you can kind of carve out this space of food that is wholly yours. And if you look kind of into like the Perfect Day patents, for example, it's they have patented almost every single mammal's milk. I think they even include like kangaroo milk in their patents. I think similarly, Clara Food patented a variety of different animals' egg proteins. So it's not just chicken eggs. No. When you have the opportunity, you patent kind of almost every single variant. So I think that's like a really interesting space where people are identifying different proteins and protein groups that can be made at scale using these kind of production technologies that exist, and kind of making that at scale. And so I think fermentation, if you're really interested in going to the space, fermentation is probably where there's like a lot of interesting develops that can be done, because a lot of the tech has been de-risked and developed already. And so it's just about picking interesting products and producing it at scale. So opportunities there where people can work on different things is really focused on scaling. So bioreactor design, bioreactor conditionalities are super important if you want to produce things at scales. And there's a bunch of different people who are looking into this, but that's kind of more of like a chemical engineering, mechanical engineering question about how you have good containers and components to produce things on a continued basis. Another space is kind of just developing interesting collaborations. So there's people out here who have different companies and production facilities that can produce things. And there's a bunch of different smaller startups and companies that are interested in producing products. So making that process pretty seamless and connecting them, I think will have like a lot of interesting impact that hasn't really been done as much. The other is kind of like providing supplementary services for people who are making fermented products. So there's a variety of companies, for example, I'll highlight one that I really like called Culture Biosciences that make it, they call it like bioreactors in the cloud. So they have facilities where there's a bunch of very small bioreactors where you can just send them a microbe and then they will give you a detailed analysis of what's needed for you to actually scale up your production. And so you can make estimates on whether or not things work. So making the process for people who are interested in making fermented products really simple and seamless, like something like a cloud service or AWS, something for biological equivalent, is a space that potentially has a lot of impact going ahead in the future. Yeah, so I just talked a bunch. I could keep going, but I kind of want to hear what you guys think about that. And yeah.
I did want to give an overview of the course of what else we have going on for IAP. Because somebody else also asked, could you give an overview of what's happening in the IAP class? So for those of you signed up for the intro class, which I will remind is unfortunately only open to current students, not just at MIT, not just undergrad or grad, but any student at any university or high school. I think we have a middle school student that signed up, which is exciting. But that course is for you, and it's going to happen for January, Mondays and Wednesdays from 1 to 2:30 p.m. Eastern Standard Time. We're also going to have our guest speaker seminar series that's happening on Fridays from 1 to 2 p.m. And our first guest speaker is going to be Jeremiah Johnson of the New Harvest nonprofit that we talked about a few times in this class. And if you have any questions, please contact us at cellagorganizer@mit.edu. That's the same email that you used to sign up for this class. Sangeeta, I don't know, I might need to share my screen to show the syllabus.
Go ahead, yeah, you should be able to.
Let's see how it goes. So for the syllabus, we have today where we're giving an overview of the whole field. On Wednesday, we're going to talk about cell lines, how we, from like isolating them to making them immortal. How do we make sure that they don't change over time? And then we're going to start working on our final project, which we are really excited to roll out. Then on Monday, we're going to talk about media and also start planning our final projects. Wednesday, we're going to talk about scaffolds of next week. And then on week 3, we're going to talk about bioreactors. And on Wednesday of that week, we're going to talk about downstream processing and regulation. Then on the last week, we're going to talk about food science. We're going to wrap up our final project, and on Wednesday, we'll present our final projects in a sort of friendly competition for some Brave Robot ice cream or Perfect Day ice cream. Yeah, so that was a brief overview. Feel free to keep asking your questions or giving us what you think fermentation should be called other than fermentation, or cultured meat should be called other than cultured meat.
I like clean meat a lot.
Clean meat? Yeah.
Yeah, it's like it's a little less specific, but I think it's super marketable. And I kind of agree that like technically all meat is cell-based meat because it's
Mhm.
based on cells. So I guess I'm not a big fan of like that little nuance
Yeah.
for cell-based meat.
Yeah, I think clean meat is definitely a good one. It's sort of, I think about how there's sort of the names for cultured meat that you can see that the person thinks about it positively, and the names for cultured meat where the person saying it thinks it's kind of weird. So clean meat is one of the, like, this is a better option. I think this is better option than what we currently have, versus, I don't know, when people say things like synthetic or weird meat, or lab grown, even, that is sort of new and weird and novel.
I was listening to
Jiggy meat.
I was listening to a podcast a while ago. I forgot what it was from, but before the company just started working on stuff, if we could just call it Just Meat, one, because it was literally just meat, but two, because it was more sustainable,
That's
more
true.
ethical. Yeah, I like that one.
Have they like trademarked that name?
Yeah, I don't know which. I don't remember if I heard it first or like, I don't even remember what, or might have even been the same people. I'm not even sure.
It's
Yeah.
interesting because also like in all of the publications that you'll see about their launch in Singapore, a lot of it goes by lab grown meat. Like, oh,
Hmm.
lab grown meat was approved or verified or whatever. I'm less of a fan of lab grown meat, but
Yeah. So it's not like actually grown in a lab. Like, as it, when it grows at scale, it's not going to be in a lab. It's going to be like in a sort of brewery or carnery, as you might have it.
Exactly. That's why I'm a really big fan. I think somebody else said it too, but craft meat.
Yeah.
I love that. I love that because it also makes it sound like a little like, oh, it's like artisan almost.
Yeah.
Like
Yeah.
the downstream processing. There's so much freedom you have there in terms of how you build up the final product, and so there's a lot of artistry that actually does seem to go into it. So craft meat.
Absolutely. Yeah, and when you talk about even, I think that would be cool then to talk about the enhanced meat, or like the meat that isn't really mimicking conventional meat, but it like not just vitamin A enhanced, but it could have like a more umami flavor or taste like a meat that you've never heard of. Yeah, definitely.
The possibilities really are endless, which is so exciting.
Yeah.
I guess
I
that's
love
somewhere that's
Andrew's
a little
work.
in between. It's a little in between the science and the entrepreneurship. It's like the design, like almost like meat design, like or just what
Yeah.
you're doing with like the direction of the actual final product itself, but almost less even looking at consumers directly and just like what's a cool thing we can make that people never been seen before, but people would actually potentially eat.
Oh man. Yeah. Have you ever heard of the in vitro bistro? Because it's sort of that design element, but it's super weird and you might like it, but other people might be really grossed out by it.
I just
Thanks.
like looked into the website of that and it was so cool. Like they had a lot of sort of like the fancier meats and things that like caviar and high end sort of luxury foods. So it's really cool that there's like even a market for that.
Yeah. They also have things like cubes made from your favorite celebrity cells. Or like you can they have the pig in the garden, where like they will have they imagine that you would like have the pig in the yard next to the restaurant, and you can like choose which pig you'd like your sample to come out of. Yeah, sweetbreads, teppanyaki from the aquarium. Yeah, all sorts. Oh, meat grapes. I don't know. There's also a video that I've watched where people have made meat in grapes, as we saw earlier, that like you can decellularize plant material and it has natural vascularization. So having meat fruit would be also pretty cool. Yeah, there's a lot that you can do once if you are not really as interested in talking about conventional or like our cultural ideas of meat, but going into the unknown.
What do you guys think about cannibalism in cell ag? I've read a serious article about it, and there was something about a kit at home where you could just take some of your own cells and then grow meat from it and then eat it.
I heard of people, I think this was actually at Ginkgo Bioworks. The creative director grew cheese from the bacteria that grew in like between her toes. I don't know.
I think there was also an art project that was made, I think like 5 years ago, where someone was working on auto cannibalism, where they were taking samples of themselves and doing it as a, gosh, I'm forgetting the name now, but it's definitely a thing that you can do. I don't know exactly why you would want to, but
Yeah.
yeah, I guess another thing you kind of have to consider like a bit more seriously when you are talking about that is the potential for passing on diseases and how to monitor and regulate for that. So usually when you talk about cell lines for meats, it's kind of been well prepped and established, and you can screen for any issues, right? If you talk about like, for example, like zoonotic diseases, you know, all this stuff that we're worrying about now with, for example, coronavirus, you can, you know, monitor for that in a very detailed manner. And it's much harder to do if you are kind of taking samples from the goal. It's an interesting idea, I guess.
Yeah. Sangeeta, do we have a question? What podcast were you listening to?
Oh, I thought I had put in there. It was the Culture Meat.
Oh,
and
okay,
Future
yeah,
Food.
yes, that, yeah, the Cultivate, the
That's a good
Culture
one.
Meat and Future Food podcast show is, yeah, like probably maybe the first podcast that's like mostly devoted to this field. Yeah, it's super cool. They've interviewed a number of both like industry and academic experts. Would it be possible to cultivate cells from animals that have already gone extinct? Yes, to some extent. This also like gets into de-extinction efforts. It will partly depend on if you have a source of complete DNA. Like how long ago did that animal or plant or whatever go extinct? So there have been efforts, for instance, to de-extinct the woolly mammoth. So that might not be from tissuing cells directly, but you might make some changes to the ancestors of that species to revert it back to the mammoth cells. Yeah, so that has been done before in the Church Lab, I believe. They made tissues of mammoth, and they also didn't think about it as cultured meat, but just as this sort of de-extinction effort. Yeah, people are definitely thinking about it.
I don't think we have the DNA sequence for a T-Rex, do we? It's
Yeah,
also,
that
that's
went
very
extinct a
Jurassic
bit
Park.
longer ago.
Yeah.
Yeah, we could probably have dodo omelets. That would probably be easier to get a DNA sample from.
Yeah.
Um,
Elizabeth.
So yeah, the Zoom link for the rest of the course will be sent out. I'll send it out later today. But we had to make a separate emailing list because we didn't want to spam those who were unfortunately we unfortunately cannot take in the intro course, as that one's only, as I said, current students. Yeah. All right, it's 3:04. I can
Thank,
we can
thank you
stick
guys
for
so
the
much for having
questions.
this.
Thank
This
you.
was crazy information dense, but looking forward to the class this month.
Yeah.
Yay.
Thank you
unpack,
so
unpack
much.
some of that.
Thank
Yeah.
you.
Yeah.
Thank you
you,
for being
everyone.
here.
Thanks, everyone.
Yeah. Have a great rest of your day.
Thank
You
you.
too.
See you
Yep.
later.
Thank you and bye.
Thank you
See you
guys.
Wednesday.
Great. Do
I'm
you
gonna
all
stop
have
the recording.
questions?
Yeah, you saw.
Yeah.